everyone. It is my pleasure to share my lab's research journey with you in the Stellenbosch University public lecture. I wish to acknowledge a whole team of clinicians, researchers, and students and collaborators. There are too many to name all here, and I will acknowledge them individually in the presentation where applicable. However, I need to acknowledge Doug Kell, my long-standing collaborator. We have been collaborating since 2011. Our lab's research will not have been possible if not for the amazing funders, Polybio Research Foundation, and in particular, Amy Prohl, Balvi, and Kandru Research Foundation, and the amazing Vitalik Buterin, who contacted me personally early on in the pandemic. His and his team's support amazes me every time I think of them. Colonel's crowdfunding initiative, and also the thousands of clinicians, researchers, and patients who made it possible for us to obtain an imaging flow cytometer in our lab. And also Standing Up to POTS Foundation and the South African National Research Foundation and the MRC. Over the years, we have published numerous papers that show the importance of inflammatory molecules in circulation and their role in abnormal clotting. I'm particularly interested in platelet signaling and the role in abnormal clotting. Platelets in circulation play a critical role in healthy blood clotting. However, they can become overstimulated and can drive pathological blood clotting if there are inflammatory molecules in circulation. This can also happen in the presence of viral and bacterial infections, where platelets can act as important signaling entities. There is also a complex relationship between receptors on platelets and endothelial cells where circulating inflammatory molecules may bind to. In the context of COVID-19, platelets are therefore central in immune activation and general coagulation pathology, and they can form various complexes and obviously also with themselves. This is known as platelet clumping. We are also interested in pathological blood clotting involving the main clotting protein, fibrinogen, which is a soluble protein in circulation. Together with Doug Kell, we discovered that these soluble fibrinogen molecules may become insoluble in the presence of circulating inflammatory molecules and may then take on an amyloid structure when they interact with inflammatory molecules or inflammagens from viruses and bacteria. Also spike protein from SARS-CoV. We have shown this amyloid presence in blood plasma using various amyloid protein markers like theoflavin T and amitrackers. Theoflavin T is a well-known marker for amyloid protein and has been used for many years to study amyloid proteins, in particular amyloid beta, in the brains of Alzheimer's patients. We call these structures fibrinogen amyloid deposits. As I will discuss later, only in 2021, we started using the term microclots. Here is an example of our earlier work on ischemic stroke. The green signal is that of our marker for misfolded protein, theoflavin T, and it demarcates the amyloid areas in the fibrin. In the micrograph on the left, in A, is an example of blood plasma from a healthy individual with no visible amyloid protein misfolding. And the micrograph on the right shows plasma from a patient who had a thrombotic ischemic stroke. Significant amyloid plasma protein misfolding is visible. Between 2014 and 2016, we discovered that the potent inflammagen that comes from bacteria called LPS can directly bind to the plasma proteins and cause them to become amyloid. Here are micrographs of our earlier work. On the left, you can see plasma from a healthy participant with minimal green theoflavin T signal. And on the right in B, there is LPS that was added in very low concentrations. Significant amyloid formation could be induced and was now noted. This laid the foundation for our later work on acute COVID and then also long COVID. We also studied clotting pathologies in various inflammatory diseases by adding thrombin to the blood or blood plasma. Thrombin is the last step in the clotting cascade that transforms the soluble clotting protein fibrinogen to a little insoluble gel or fibrin clot. It is well known that this is a lab method to study how fibrinogen clots into this fibrin gel. In the micrograph in A, you can see a healthy fibrin net created from plasma of a healthy individual. In healthy individuals, this fibrin net clot looks like a little fish net, but in diseases like diabetes, cardiovascular disease, or even stroke, the fibrin clot becomes dense and matted and clots into deposits, as can be seen in micrograph B and C. This clogs up the vasculature and can cause significant pathology. We were also interested in the structure of red blood cells and their death mechanism, called eruptosis. On the left, you can see a red blood cell from a patient with severe iron overload disease, known as hemochromatosis. Too much iron in circulation causes oxidative stress, resulting in a significant inflammatory process. On the right is damaged red blood cell entrapped in fibrin from a patient with thrombotic stroke. In 2014, we were the first researchers to show that in Parkinson's disease, there are significant red blood cell eruptosis. This we directly attributed to circulating inflammatory molecules. At that stage, the idea of a microbiome in blood was still a new concept. We argued that inflammagens, like LPS, from bacteria in circulation, could also directly cause such pathologies. Now it is widely accepted that there is indeed a blood microbiome and that it has significant impact on pathology as well as disease progression. Fast forward to the pandemic. Let me now take you on an incredible, difficult, but satisfactory journey since 2020. Early on in 2020, we looked at blood clotting in acute COVID-19 using scanning electron microscopy. We found that platelets were damaged and clumped together and attached to red blood cells. The fibrinogen and clotting proteins that is supposed to be soluble form spontaneous deposits. In the micrographs on the far left, you can see platelets that are hyperactivated. Note the yellow arrows. In the middle are micrographs where platelets are attached to red blood cells. Note the white arrows. On the far right, you will see micrographs of spontaneously formed plasma protein deposits that even glue together red blood cells. Note the blue arrows. During the pandemic, we did not have access to human thrombin. Thus, I decided to only add theophlavin T, our amyloid protein marker, directly to platelet-poor plasma. Here you can see the differences in platelet-poor plasma protein structure in acute COVID-19 patients where we compared plasma smears of these acute COVID-19 patients to that of healthy and diabetic patient plasma samples. The insoluble amyloid plasma proteins are once again demarcated as the green signal from theophlavin T, our marker for misfolded protein. As we now expected, we found small areas of theophlavin T signal in plasma of healthy individuals, as can be seen in the micrographs on the left. More abnormal amyloid deposits were noted in plasma of individuals with diabetes. What was, however, quite significant to us was the extent of the abnormal deposits in acute COVID-19 plasmas, seen in the far right plate. Also note the scale of 10 micrometer at the bottom of the micrographs. In 2021, we also studied the effect of the addition of S1, part of the spike protein, on clotting when we added it to healthy blood samples. Here you could see fluorescent microscopy micrographs where S1 was added to healthy blood samples. We could again, as we could previously with LPS, directly induce amyloid protein misfolding and significant platelet hyperactivation. See the micrographs on the left, suggesting that the spike protein also has direct protein-protein interactions with fibrinogen. However, the extent of the amyloid formation was extensive. S1 also activated platelets via binding to platelet receptors. See the micrographs of the platelets on the right. If you turn your attention to the scanning electron microscopy results of this experiment, on the left, you will see red blood cells of a healthy individual and A, and in B, a healthy platelet. Major clumping of plasma protein after the addition of spike protein S1 can be seen in C to H. We also initiated a microfluidics experiment. See the plate on the right. Healthy plasma have few plasma deposits. Note figure A and B. However, in acute COVID-19, and this is of note, is the earlier Delta variant, significant plasma deposits were seen flowing in the fluidic system, seen here in C and D. We could also induce these insoluble plasma deposits by adding S1 protein to a healthy plasma, CE and F. Now we had evidence that spike protein is directly involved in the clotting pathologies seen in acute COVID-19. Now, with our knowledge of acute COVID-19, end of 2020, we also turned our attention to long COVID. Early in 2021, the term microclots were coined to describe the insoluble amyloid deposits we found in both acute and long COVID. We looked at blood samples from patients and soon realized that there are persistent plasma deposits and widespread endothelial dysfunction in these patients too. We hypothesized that these clotting pathologies may be central in causing the widespread symptoms in these patients and may lead to tissue hypoxia. Long COVID turned out to be a global catastrophe. It still is. It is now estimated that there are 400 million cases worldwide and counting. Conservatively, between 10% and even as many as 30% of individuals who have had an acute infection continue to have persistent, debilitating symptoms after the active infection has cleared. It is now well recognized that long COVID results in widespread organ damage from the heart to the immune system and clotting pathologies, and most worrying, significant brain pathologies. Here are representative examples of our earlier findings. Here is an example of a red blood cell of a healthy individual on the left. On the right, significant microclot formation in long COVID blood samples were noted, where these microclot deposits also, again, just as with acute COVID, glue together red blood cells, suggesting significant impairment of the function of the red blood cells, which is mainly carrying of oxygen to all organ systems. Here is a close-up of these deposits on red blood cells. Again, a larger magnification of a red blood cell, showing significant microclot deposits on the red blood cell membrane. This will most definitely impair red blood cell flow capacity and their ability to transport and deliver oxygen, but also remove CO2 in the capillary beds. Here you can see the spontaneous, dense, matted fiber net formation in blood from a long COVID patient. Nothing added to it, straight from the tube. Again, theoflavin T lit up significant amyloid microclots in long COVID samples. Here you can see a huge clot from someone that suffered from the disease. Turning to our proteomics research, in 2021, we published our first proteomics long COVID paper. As I previously mentioned, the term microclots originated from this proteomics paper. I was interested in the protein footprints of acute and long COVID samples and used blood samples from healthy and diabetes samples pre-pandemic. To our surprise, we could not digest the plasma of the acute and long COVID samples with a standard laboratory trypsin digestion method. Our biochemist Marie Flock developed a double digestion method, and we found numerous entrapped inflammatory molecules and also antibodies inside the digested microclots. We found a molecule that is involved in blood clotting and in endothelial damage called von Willebrand factor, also CRP and serum amyloid A, also the various fibrinogen chains, as one would expect, but also alpha-2 antiplasmin. Here is the clotting cascade. You can see here where thrombin is found in the cascade as the last step in forming the little fibrin clot. If clots form in the body, they must be broken down. The process is called fibrinolysis. Of particular interest was where alpha-2 antiplasmin might act or interfere. This molecule prevents clot breakdown. Now we had our answer why these microclots might persist beyond acute infection and why they break down much slower than one would expect. They entrap molecules that interfere with normal clotting and this clotting breakdown cascade. Later in 21, we also completed a larger proteomic study where we looked at 100 long COVID and city control samples. Some of our most noteworthy findings from this paper include a downregulation of kallikrein with an upregulation of platelet factor 4 and again von Willebrand factor, and also a marginal upregulation of alpha-2 antiplasmin. If you turn your attention to the clotting pathway diagram again, these molecules may play significant roles in this clotting pathway and again points to a decreased fibrinolysis and eventually a fibrinolytic failure. We also found significant presence of antibodies and even autoantibodies entrapped inside the microclots. Our findings again suggested that hypercoagulation and vascular damage are key role players and key in causing the various persistent symptoms. Following on from this research, we again revisited our stored plasma samples to look at levels of the inflammatory molecules in the soluble fraction of the blood. We plan an experiment to look again at von Willebrand factor, platelet factor 4, serum amyloid A, and alpha-antitryplasmin. We also included E-selectin and PCAM2, well-known endothelial damage markers. We found these molecules were also significantly upregulated in the soluble fraction of the plasma. We used standard ELISA laboratory methods. This and our proteomics analysis therefore suggested that these molecules are present in two fractions of the blood, either entrapped inside the microclots in significant numbers or present in the soluble fraction. Of significance might be that if a pathologist would measure only the soluble fraction using standard laboratory methods, the entrapped fraction would not necessarily be taken into account. Early 2022, our research entered the international arena. In February of 2022, a US government document, the GAO, that directs important research focus areas, was published. Microclot presence, viral persistence, and autoantibodies, together with organ damage, were recognized as central pathologies to look into if we needed to find answers for long COVID. Our work was also featured in June of 2022 in Science, also in Nature and National Geographic. In 2022, it became clear that we needed an easily accessible method that could be used in general pathology labs. I turned my attention to flow cytometry, but soon also realized that we will have to convince clinicians and researchers that microclots are not only cell debris to be ignored. For that, we needed an imaging flow cytometer. With the help of Balvi, Polybio Research Foundation, and kernels, we had our instrument within 6 months. 6 months later, we published our first imaging flow cytometry method with the hope that it would be a useful diagnostic method to quantify microclots in plasma samples. So why is it important to determine if microclots are present in circulation or not? We and others believe that microclots may result in ischemia, reperfusion injury. So what exactly is ischemia reperfusion injury? It is initiated via bursts of reactive oxygen species produced during the reoxygenation phase following hypoxia. And this is a well-known process in a variety of acute circumstances. We argue that this type of injury also underpins elements of the pathology of long COVID. Ischemia may be initiated via fibrin amyloid microclot deposits, thus temporarily blocking capillaries, for instance, as exercise is started. Reperfusion is a necessary corollary when it finishes. Such microclot-based phenomena resulting in ischemia reperfusion injury can explain both the breathlessness, the fatigue, and the post-exertional malaise that may be observed in long COVID, and ischemia reperfusion injury might therefore explain many, if not most, of the various organ damage and symptoms seen in this diagram and that patients complain about. In 2023, and also again in 2025, together with a larger international collaborator group, we published 2 papers on the possibility of viral persistence. Viral persistence might give us a reasonable explanation for the constant shedding of spike protein into circulation, resulting in persistent clotting pathology, platelet hyperactivation, and widespread endothelial damage. This will ultimately result in systemic thrombotic endotheliitis. I was also involved in the writing of a Delphi for long COVID. This modified Delphi electronic survey on long COVID was conducted under the governance and oversight of the World Health Network. Researchers and clinicians of 28 countries participated. This Delphi paper forms initial guidance to address the spectrum of long COVID as a disease and reinforces the need for translational research and large-scale treatment trials for treatment protocols. Together with a team of clinicians, we also wanted to give some guidance to clinicians on how to determine if their patients indeed have thrombotic endotheliitis. We wrote a paper with title, Vascular Pathogenesis in Acute and Long COVID, Current Insights and Therapeutic Outlook. It was published in Seminars of Thrombosis Hemostasis in August 2024, and it won the most popular paper for 2024. Here you can see the team of collaborators, Dr. Arne Creer from South Africa, Dr. Dave Chaffee from Australia, Dr. Graham Lloyd-Jones from the UK, Dr. Asad Khan from the UK, Dr. Spila Salomon from Austria, Dr. Jaco Loubser from South Africa, Professor David Petrino from the US, and Professor Douglas Kell from the UK. So returning to microclots and their content. It is not only simply the size and the numbers of microclots present in healthy versus long COVID, or for that matter, any other disease with circulating inflammatory molecules, but their content, meaning the inflammatory molecules of interest, their activity, and their biochemical characteristics. We have recently been looking at the possibility that there might be various microclot phenotypes, specifically as the various molecules that us and others have found entrapped inside microclots may also have their own amyloidogenicity. By using a website called Ameliogram, the amyloidogenicity of any protein can be determined. Here is an example of alpha-2 antiplasmin, the molecule I referred to earlier. Almost all of the inflammatory molecules we have found entrapped inside microclots have a huge amyloidogenic potential themselves, making them sticky and thus prone to attach to the microclots as they roll over damaged endothelial layers. Using the Ameliogram site, this diagram can be generated. On the x-axis is plotted the amino acid residue number, and on the y-axis is plotted the amyloidogenicity score, which is essentially the probability that a given sequence segment of your protein of interest can form amyloid structures. Following on from our work in microclots, we wish to characterize the heterogeneous nature of fibrinaloid microclots into various phenotypes. We suggest that there are various phenotypes depending on their structure. Examples are where fibrinogen aggregates and form the main component of the microclot, resulting in a true fibrinaloid microclot. Fibrinaloid microclots associated with nets or neutrophil extracellular traps, possibly can form immune complexes or fibrinaloid microclots seeding onto cellular debris from damaged blood cells or blood cell membranes. They might also be a phenotype resulting in heterogeneous inflammatory molecule aggregates, which aggregate together with fibrinogen, seeding onto blood cells and endothelial cells that are not necessarily pieces or debris, but still blood cells in circulation, intact endothelial layers, which might in fact be damaged already. One such important phenotype I mentioned was microclot immune complexes. Recently, with a collaboration with Prof. Alain Thierry's lab from INSERM in France, we conducted some research. He is specifically interested in net formation and neutrophil elastase, melioperoxidase, and also circulating DNA in long COVID versus control samples. We confirmed that nets are entrapped and structurally associated with microclots from long COVID patients. Here we used the stain cytostain, which is the red stain. We could also use a stain called Huscht. So cyto, red, and Huscht are very similar, and they are well-known DNA stains. We also used theoflavin T for amyloid protein areas in the microclots and melioperoxidase antibody, which is the light blue areas. One of the questions that we frequently get is whether microclotting is related to macroclots seen in thrombotic stroke. Circulating back to our research many years ago, we found significant microclot deposits in blood samples from acute thrombotic ischemic stroke. This research was done in collaboration with the neurologist, Dr. Vibram Duin in Pretoria. At that stage, we were more interested in red blood cell pathologies and also did not yet call the plasma deposits microclots. Now looking back at our data, the microclots were hiding in plain sight. I'm happy to report with Doug Cullen, a team of researchers and neurologists from the University of Liverpool, we have in press a paper in the journal Stroke showing that retrieved macroclots from patients that had a thrombotic stroke indeed contain significant amyloid protein as demarcated with theoflavin T. This suggests that microclots are on the biochemical and physiological pathway to formation of macroclots, as seen in stroke, suggesting that microclots lead or can lead to macroclots under specific inflammatory and pathological conditions. We have just pre-printed also a paper with a team of Stellenbosch researchers funded by the South African Medical Research Council with Professor Marita Kotze and team. We look at proteomic signatures of post-vaccination, post-infection syndrome that we call PVPIS, and discussed insights into immune dysregulation and coagulopathy. This research was done with my wonderful MEC student, Maxine Waters, as first author. We compared the protein signatures with previously published data from the proteome analysis of long COVID patients and found that individuals with this syndrome had distinct protein footprints, suggesting that vaccine injury might lead to a different physiological pathway activation. Other research groups have also shown utility for microclots as diagnostic marker. Here is a fantastic paper by our collaborators from the Wust lab, where they found amyloid deposits in muscle using our theoflavin T marker in their methodologies. These markers are shown here in the green dots. This is where amyloid deposits are found inside muscle biopsies. Caroline Dalton from the University of Sheffield Hallam also showed increased microclot counts in a UK long COVID cohort. The Yonker group from Harvard also found evidence of microclots and activated platelets in pediatric long COVID patients. In a Blood Advances paper from the Toe lab in the UK, the team used our amyloid marker theoflavin T and proteomics to look at microclot composition in sepsis. You could just imagine that we were very happy to read their conclusion, and I quote, Overall, we believe that this is an important step forward in the potential utility of this assay as a clinical, useful, and practically measurable biomarker to identify sepsis, predict DIC and mortality in the ICU. A team of radiologists from the University of the Free State have recently found abnormalities in the carotid arteries that are atypical. This team is currently investigating the phenomenon more closely. You can see the fluffy deposit in the carotid artery in this micrograph kindly provided by the team. In a recent paper by Peluso and Deeks, where they discuss mechanisms of long COVID and the path towards therapeutics, published in Cell, they focused on the vasculature and its dysfunction. They suggest that the main mechanistic targets should be viral targets, immunological targets, clotting targets, as well as the human herpes virus as a target. We desperately need well-structured clinical trials. We need to characterize the patients group before enrolling them in such trials. Our laboratory focus is to understand which biomarkers are of importance in long COVID. We wish to also understand the presence of various symptoms and understand which biomarkers are indicative of which symptoms. Eventually, we would want to see that clinical interventions are customized based on a combination of symptoms and reliable biomarkers for specific long COVID phenotypes. In clinical trials, we also need measurable biomarkers before and after interventions. We believe we have a good candidate, and with the added benefit, it also entraps various different inflammatory molecules, making it a relatively easy method to quantify and track molecules of interest for clinical trials. So what are we doing further to make the research more accessible for patients and clinicians? We have patented our methods, and we are partnering with clinics and labs all over the world. Our startup company, Biocode Technologies, are in the process to develop a novel flow cytometry method too, that will go far to have standardized techniques available globally. One such collaboration is with Innes Laboratories in Switzerland. They are currently doing the microclot tests on their imaging flow cytometer for clinical utility. Together with pathology lab collaborators Yannick Koch and Felix Korkman from Innes Laboratories and the University of Zurich, as well as Alain Thierry from INSERM in France and Doug Kell from the University of Liverpool, we are closely looking at the phenotypes and the nomenclature of the term microclots, so watch this space. What will be happening in the near future? We have numerous significant collaborations and signed material and data sharing agreements with various international laboratories and collaborators. We are nearly ready to submit a paper with the Fahy Group from the University of Derby. We have some exciting new data that might craft a way for purpose-built interventions to support people with long COVID that moves us away from the more traditional rehabilitation approaches to optimize patient outcomes and safety. We have a grant with Jeroen den Dunnen, and we have optimized the microclot imaging flow test in his lab in April of this year. We will be using full body PET scans to detect microclots in patient organs. We have completed analysis of microclots from samples collected from the Robwust lab. We are in the process to write up the results. We are also far along with the analysis of POTS samples from Professor Satish Raj from the University of Calgary. We are very thankful for the Standing Up to POTS grant to do this research. This grant will also cover the funding for samples that we have received from Merel Hellemans from Erasmus University of Rotterdam. We have data ready for a follow-up study with Alain Thierry from INSERM in France. In the near future, I will also be receiving samples from UCFS and the Peluso team, and also from Bettina Hoburger from the University Clinic of Erlangen in Germany. This is just a few of the current and near future endeavors in our lab. Our aim is to develop novel methods to be used by researchers in laboratories. For that, I have a fantastic team of dedicated postgraduate students and postdocs in my lab. These students are absolutely amazing. Then, never to be forgotten, the patients that participate in our research. You are the reason we keep on doing what we do. And with that, the trip through our research endeavors have concluded. Thank you for your attention and see you soon at a next webinar.